I'm finding you.
The lecture has started.
Hi, can you all hear me? Can you hear me?
I can hear you, Mr. Jeff.
Awesome. Should I go ahead and get started, or should we wait for see if there's anyone else?
Let's just wait for a few minutes.
Sure, sounds good.
Mr. Jeff,
Yep.
we can start.
Okay, sounds good. So hi again, everyone. So yesterday we talked about how microbes grow and reproduce. And today we're going to talk about what they actually do aside from that. Okay. So again, I've lined out what I think are the key concepts. So these are some topics that I think that you would probably want to know if there's a test or anything. And I'll also mention, as with yesterday, I will be sharing the video and these slides, so you don't need to write everything down. Okay, so the first thing we're going to talk about is energy in bacteria. And so I just wanted to first review several important basic concepts. So the first thing is, what is metabolism at all when we're talking about microbial metabolism? And so essentially metabolism is just the chemical processes that are happening within a living thing in order for it to maintain life. We can break down metabolism into two different types of metabolism. One is catabolism, so that's breaking down large molecules. For example, like if you have food molecules, like something like starch, that's a big molecule. You want to break that down into sugar so you can directly use that sugar for energy. That's an example of catabolism. On the flip side of the coin, anabolism is the opposite. So that's combining small molecules to build larger ones. For example, in the cell wall, peptidoglycan is a really complex, large molecule. You need to build that up in order to have the cell wall. Another example is protein synthesis that we'll talk about in a minute. That is an example of anabolism. You're taking a small molecule, single amino acids, and you're combining them to build protein. This is all part of a cycle, and important to note is that the breaking down of molecules through catabolism often is used to generate ATP, and that's essentially the energy source of the cell that we'll talk about more in a moment. I don't know if you've learned about this in some of your other biology courses, but I want to highlight this important concept called the central dogma of biology. And this is really important for what we're about to talk about next with protein biochemistry. Basically, what's important to understand in the cell, you have genes that are made of DNA. The DNA is transcribed into RNA, which is essentially like a messenger molecule. DNA is double-stranded. RNA, as you can see, is single-stranded. DNA is made of the four bases A, T, C, and G. In RNA, there's no T, and instead there's a U, uracil. And then the RNA, once that's made, is translated into proteins. And so, there's, again, hopefully you've learned about this, but there's this, the way the code works is there will be 3 letters, so like AGU on the RNA, that's going to translate to a single amino acid, which makes up the protein. And so the central dogma is that, this is how it works. DNA goes to RNA, goes to protein. One of the interesting things I always find with biology is that every rule has some exception. And the important one here to understand is RNA can be turned into DNA through a process called reverse transcription. Some of you might remember from yesterday's lesson, we talked about HIV. So that's a retrovirus. And so what it does in the cell is it has its RNA be turned into DNA that can then, again, go all the way through this process. Okay, so we mentioned that basically, in general, the function of genes is to have them be transcribed and then translated into proteins. And proteins are made of single amino acids. There are effectively about 20 of these that most proteins use. Here are some examples here. These are just the 3-letter codes. This is phenylalanine, leucine, serine, cysteine. And so proteins are essentially, and this includes proteins in foods we eat and everything. They're essentially just a long chain of these amino acids in a different sequence. And so that's all genes are, is they're encoding for these different amino acids. But what's important is the sequence of these amino acids determines the shape of the protein. And so this is what we call primary structure. That's essentially just what is the sequence of these different amino acids. When we look at the next level, secondary structure, so here we can sort of look at the chemical makeup of these individual amino acids. They form these secondary structures that are called either beta sheets or alpha helices. So you can see how the sheet, they kind of just form basically this flat part, where a helix, it kind of turns around like this. And so you'll often see proteins be represented with these structures. And then finally, and the most important part, is these secondary structures kind of fold in on themselves. And it's all driven by essentially the fact that in this aqueous water environment, the water basically pushes the molecules together in a certain way based on the chemical properties of the individual amino acids. And you end up with what is called a tertiary structure. So that is what the protein actually looks like in 3 dimensions. Now that's important because of the way proteins work that we'll talk about next. Now some proteins are structural or have other roles, but when we're talking about metabolism, the type of protein that we really care about is called enzymes. So you've probably heard this word before. And essentially an enzyme is just a type of protein that catalyzes a chemical reaction. And so here's a diagram of what that might look like. So you have your enzyme, and it's folded in a certain way, and based on the shape of its folding, a certain substrate molecule can fit into this pocket. And then once it's in there, the protein, the enzyme can enable a certain chemical reaction to happen. In this case, you see this is like a hydrolysis reaction. This is a breaking up of this initial substrate into 2 separate products. You can certainly have enzymes that do the opposite of this, where it would take in two substrates and combine them into a single product. This is a example that you might see in the microbial world. So we have maltose, which is two glucose molecules, two sugars connected. And then you have this enzyme that breaks it down into two single glucose molecules that can be really easily used for energy. And so this would be an example of catabolism. And the way you can remember the difference between those two is that cats like to break things. So in microbes, there's a lot of different strategies for where they get their energy. And essentially, there's two questions you want to know about understanding the metabolism of an individual microbe. One is, what sort of carbon source do they use? Some microbes can do what's called fixing carbon from carbon dioxide or other inorganic molecules. And those are called autotrophs, if they can fix their own carbon. Alternatively, many microbes need organic compounds, so things like sugar, and in that case, they will be a heterotroph. The other question is, what do they use as their energy source? Some microbes can use light in photosynthesis, and then they'll be called phototrophs. Other microbes can use chemicals like metal molecules, and those are called chemotrophs. And so essentially when you combine these two, for example, if we look at E. coli, E. coli uses organic compounds. It can't fix its own carbon. And it cannot photosynthesize. So it is a chemoheterotroph. I mentioned briefly ATP. If you've studied mammalian cell biology or any cell biology, you've probably heard of this molecule. So it stands for adenosine triphosphate. And it's been described as the molecular unit of currency. It's like money in the cell. And it's a way to store energy. So if you look at the chemistry of it, this part here is the adenosine, and then each of these is a phosphate group. So adenosine, 1, 2, 3 triphosphate. In particular, there's a lot of energy stored between the second and third phosphate bond. And so this is a great way for cells to store energy when they need it by creating this ATP, and then when they break down this bond, they use the energy. You can also, so if you break off one phosphate, you have adenosine diphosphate, and if you break off two, which you can do, you have adenosine monophosphate. And this is just an example of glycolysis, the breaking down of glucose in the cell. And this kind of, you don't need to fully understand this pathway, I don't think, but just understand that glucose is used as a way to make energy. So, and there's, complex chemical reactions happen. So in one case here, at this first step of the pathway, we're actually spending an ATP, turning it into adenosine diphosphate here in order to do the first part of the reaction. But other steps further on, see here and here, are ATP generating. There are other, you can see some byproducts like water that are produced. And the other thing I'll point out are NADH. This is basically it's an electron carrier molecule. So NAD plus means you basically, so it's positively charged, and then it gets an electron from this reaction, and then it becomes NADH. You can have the opposite happen, where you lose an electron. And so this is important because a lot of these chemical reactions that happen need an extra electron or to get rid of an electron. And so this molecule is really important for the cell for carrying out these what are called redox reactions. I don't have it on the slide, but I don't know if you've learned about redox reactions. So it's a kind of portmanteau of reduction and oxidation. And you can remember the mnemonic oil rig. So it stands for oxidation is losing, OIL, oxidation is losing electrons, and reduction is gaining electrons. So basically these redox reactions are really important for some of this. But the important thing here to know is just that, glucose is sugar. We use sugar for energy, so do microbes. Sugar and other carbohydrate sources can be used to generate ATP as an energy source. And we'll talk about this more in further slides, but there's some important byproducts of this that come out, such as ethanol, so alcohol that we drink. That is the result of microbial metabolism. Lactate, if you've ever had pickled foods, lactic acid producing bacteria are involved in that. Okay, so I also mentioned photosynthesis. So you're probably familiar with photosynthesis in plants. Certain bacteria, such as cyanobacteria, can produce their own energy from sunlight. And you don't need to memorize this chemical reaction, but I think basically the important thing to understand is that this reaction of taking light as an energy source can be used to generate ATP and NADPH, which is another electron carrier molecule that we talked about. Through this pathway, and if you have carbon dioxide fixation capabilities, we can use this carbon source and the light, the energy that we get from the light, in order to generate sugar, so either in the form of like glucose or pyruvate, shown here, and then we can proceed essentially with the glycolysis reaction that we saw in the previous slide. So not all bacteria can do these things, so this would be an example of a photoautotrope that can fix its own carbon from carbon dioxide, and it can get energy from sunlight and do photosynthesis. And then on this topic of understanding, photosynthesis and ATP, I wanted to talk briefly about endosymbiotic theory. This is another thing that you might have learned about in some of your previous classes. But again, if you've studied cell biology, you know that animal cells have mitochondria that are energy generating. That's where all our ATP generation in our cells is happening. It's called the powerhouse of the cell, super important for how our cells function. In plants, they also have mitochondria, and they also have chloroplasts as well. And the chloroplasts are what the plants use in order to carry out photosynthesis, to get energy from light. So endosymbiotic theory is this idea that somewhere in our evolution of eukaryotic cells like we have, those cells engulf bacteria, so either aerobic bacteria or cyanobacteria, and that those bacteria were essentially the precursors to what are now mitochondria in our cells and chloroplasts in plant cells. So this is I think, widely accepted theory. If you look at the genes in mitochondria and chloroplasts, they have a lot of homology. They're very similar to what we see in certain bacteria. But I think it's just really cool to understand and think about this fact that we have basically like little bacteria in each of our cells as mitochondria that are helping to carry out this the reaction of ATP generation that we previously talked about. Okay, any questions on that section before we move on? Okay, so then briefly, I just want to talk a little bit about nutrient metabolism. One important ecological interaction to understand is nitrogen fixation. So we already talked about carbon fixation. It's also important in many cases to be able to fix nitrogen. And this is super, super important to understand for agricultural microbiology, because plants can't do everything that they do on their own. There are microbes in the soil, mainly bacteria and fungi, that carry out certain chemical reactions for them, and interact with the roots. There's this symbiotic relationship between the microbes and the plants. And so it's really important that there's many like agricultural startup companies out there that are trying to figure out how can we design, basically it's almost like probiotics for plants, different fertilizers and stuff. How can we use microbes as a way to help plants grow? And so nitrogen fixation specifically is about fixing nitrogen, or in other words, converting atmospheric nitrogen in the form of N2 gas, that's about 70% of our atmosphere, into ammonia, which is NH3. And then plants are then able to use this NH3 as a nitrogen source to incorporate into their organic molecules, such as proteins, for example, need nitrogen. And so here in this image, you can see these root nodules are places where you have an interaction between nitrogen-fixing bacteria and then the plant roots. Okay, so then I just want to briefly talk about the types of macronutrients. So, these include fat, carbohydrates, and proteins. These are the same things we eat, many microbes can eat. Now, we need a balanced diet of all of these things. Microbes are more complicated. Some microbes can survive on like just pure sugar, for example. And so here I have, as an example, a plantain. Plantains are a little, maybe not the best example because they're really complicated, because depending on how ripe they are, they're going to have different nutritional content. So the yellow or even black plantains will have a lot more sugar. But at any rate, this is an example of a nutrition fact label you might see that shows the breakdown of what macronutrients are in this plant, including fats, carbohydrates, and proteins. My work focuses a lot on carbohydrate metabolism, and so selfishly, that's what I'm going to focus on as an example here. But it's important to understand that different microbes can work with all of these molecules. Every microbe is going to be synthesizing its own proteins, fat metabolism is also important because the cell wall, cell membrane is largely fat-based, so it's important to be able to work with those fat molecules as well. So I just wanted to touch on the different types of carbohydrates that are out there, because I think it's important to understand carbohydrate metabolism. So, if you look on a food label, carbohydrates will often be broken down into sugar and dietary fiber, which is what I study. So sugars are these simple hydrocarbon molecules. Basically, it's a ring. So a 6-member ring here is glucose. And then here, fructose, you may have also heard of, is a 5-member ring. Now, what's important about these sugar molecules is that they can link up in different ways. If you put a glucose and a fructose together, that is sucrose, which is table sugar that you've probably eaten. If you string a ton of glucoses together, you get amylose, which is a type of starch. So if you like potatoes or wheat products, or even plantains are pretty starchy. That's what you have, is there's a lot of glucose molecules linked up together. Importantly, the human body has enzymes capable of breaking down simple sugars and starches, and that's about it. But the microbes that we have in our gut have a huge diversity of carbohydrate degrading enzymes. And that allows them to break down some things like fiber. Now, fiber is essentially, it's basically complex carbohydrates that aren't degraded by the human body. So essentially that includes any sort of linkage combination other than starch. So for example, inulin. I mentioned this yesterday. Inulin is it's basically a lot of fructoses strung together with a single terminal glucose molecule. And inulin, so this is one of the things that's found in plantains, for example. Yeah, so our body can't break it down. It makes it to the gut, and then there are certain gut microbes that can break it down. Okay. I also want to just talk about, again, I mentioned this briefly yesterday, but I think it's really important to understand in metabolism that microbes do not exist in isolation in monocultures in nature. In other words, you don't just have a ton of E. coli all in one place. They live in complex communities with lots of other microbes. And so it's important to understand that there's a relationship between what are called primary degraders and then secondary degraders. So for example, in the case of inulin, these are some examples of primary degraders that might be able to degrade inulin. And then these are secondary degraders that can't degrade inulin, but can break down its byproducts and turn it into things such as butyrate that are helpful for human health. And so this heat map is essentially showing you interactions between these different types of microbes. So if you see red, that means it's a positive association. So for example, Bifidobacterium longum and A. hadris are really often seen together. So in other words, that seems to indicate, it's just a correlation, but it seems to indicate that perhaps this microbe is feeding this microbe with some of its waste products. By contrast, if you see blue, that means the microbes are anti-correlated, so there might be some sort of competition within them. But what I want to focus on is this red, this sort of positive interaction of microbes feeding each other different molecules. And then, we can even eat microbial molecules such as, alcohol. Alcohol is just a microbial waste product, but then, we can drink alcoholic beverages, and our body can use that as an energy source. Hopefully not your only energy source. The other thing I wanted to point out about carbohydrates, and this extends to other macronutrients such as proteins, is that carbohydrates can be metabolized in many different ways. You do not need to understand everything that's going on in this diagram, but just notice you have the carbohydrates coming in here. It can be converted to pyruvate, we talked about before, and that can go in this direction. It can go over here to succinate, which can be converted to propionate, valerate, all these acetate, butyrate, all these different molecules. And so I just want to put it out there that in a given cell, in a given type of bacteria, there is the capacity to produce a lot of different byproducts from its sugar. And so in the next section, we will talk about what drives those changes. But are there any questions before I go on about just carbohydrate metabolism or anything else we've talked about in general? Okay, and I will move on. Okay, so in bacteria, and this holds for all of biology, but we're going to use bacteria as an example. We have what are called molecular circuits. So how does DNA actually do all the many things it needs to do? We talked about DNA encoding proteins, but there can be more than that needs to happen. We need to not just produce every protein all the time. We need to be able to turn certain things on or off depending on the conditions. The lac operon is a great example of this in E. coli that has been super studied in depth. I don't expect you to fully understand all the nuances of it from this one diagram and one slide, but I'd encourage you to read up more on it on your own later. There's been a lot written about it. And so essentially the purpose of the lac operon in E. coli is to It's involved. It has several genes that are involved in lactose metabolism. Lactose is another type of simple sugar that's found in milk. So lactose, milk. So if someone has lactose intolerance, they can't drink milk. Importantly, these genes are only expressed or produced when lactose is present and glucose is absent. So if glucose is present, the cell wants to eat glucose. But if there's no glucose and lactose is the only sugar available, the cell will then want to switch to eat lactose. So how does it do that? So essentially, you have these several different components on the operon. An operon is basically just like a large set of different genes and regulatory elements. So RNA polymerase is a molecule that's going, so we talked about the central dogma. RNA polymerase is going to bind to the DNA and produce RNA from it that can then be used to translate into produce proteins. Now first we have this cap site. This is the catabolite activator protein, and that is essentially a glucose sensor. So it will activate transcription of the operon, but only when glucose levels are low. Okay, so that's how we're dealing with the glucose sensing, is through that element. Now how do we deal with the lactose sensing? Because if there's no purpose on turning on these genes unless you actually have lactose present. The way that is dealt with is through this repressor here. So if there's no lactose present, the repressor binds to this operator region of the DNA and blocks the RNA polymerase from proceeding and transcribing these different genes. However, if lactose is present, it effectively binds to this repressor and blocks the repressor from binding to the operator. And so then RNA polymerase can proceed and turn on these enzymes that will then be able to degrade the lactose and use it as an energy source. Again, I know this is kind of complicated. There's a lot going on here, but the main takeaway is that you have in DNA sequences these basically circuits that are able to sense different nutrients and turn genes on or off based on those nutrient conditions. Okay, so we're going to talk a little bit more about what are some of the effects of that as we go forward. Just kind of have in the back of your mind that there is complex molecular circuitry driving all of these changes. So one example, so yesterday we talked a lot about different factors that can affect microbial growth, whether microbes grow or not. Similar environmental changes can also affect not only whether microbes grow, but also what they are doing. So Saccharomyces cerevisiae, we talked about yesterday, this is yeast. So this is baker's yeast, brewer's yeast used in bread and beer, all sorts of other foods. It is a facultative anaerobe. So you might remember from yesterday that means that it can grow if there's no oxygen, but it's going to probably prefer if there is a little oxygen. And if we understand the metabolism of the cell, that's going to make a little more sense. So depending on whether oxygen is present or not, the yeast can do one of two things. It can turn on this respiration pathway if there is oxygen present, and that's going to generate ATP at these two steps. However, if there is no, so it's important to understand that this requires oxygen in order to do this. If there's no oxygen present, it's going to do what's called fermentation. Okay, and so that means it's going to produce ethanol as a byproduct and then go this way. So some of the energy here is lost by producing this ethanol, and that's important to understand. So that's why it's going to prefer to do respiration if there's oxygen, because it's not wasting energy in the production of ethanol. However, for us, you know, if we're making beer or something, then we actually want it to be going into fermentation and producing ethanol, because that's what we care about. So in that way, we can use microbes for fermented foods by turning on fermentation. And so what you want to do is make sure that it's not getting Another example that can change metabolism that we talked about a little bit yesterday is pH. So how acidic the media environment is. This is an example from my field of the gut microbiome, where people looked at gut microbes and from different stool donors. So basically they just took poop from these people, and they modified the pH of the environment. Either they started at a relatively low pH of 5.5 and moved it up to a high pH of 6.5, not high, but just like relatively high, so 7 is neutral. Or they, so in another part of the experiment, they, I think, did the opposite. But here they just show this happening for 2 donors. And so these short-chain fatty acids, so these are in some of the other diagrams, but these are just molecules that bacteria can produce as waste that are actually useful for the human body. The cells in our colon and our large intestine will use some of these, especially butyrate, as an energy source. So it's important, that's why we want to understand under what conditions these are produced. So here you can see, if we look at what's happening when we increase the pH, we're having more acetate and propionate and less butyrate. So just, at a high level, it's important to understand that, changing the pH here, and this isn't just one microbe, this is a complex community of microbes, but in this complex community, by altering the environment, we can change the function, the metabolism of these microbes. And you can see for this other donor, the same trend generally held. Another concept, and this is maybe getting a little advanced, but I think it's really interesting and it kind of gets into the classical microbiology of it all, is something called Diauxic shifts. So when you have microbes, in culture in the lab, we might have microbes have just glucose as their only carbon source, but in reality, there might be multiple carbon sources available, such as glucose and fructose. And what you might expect, so if I'm as a person, if I have a plate of food in front of me, I might just kind of eat it all at once in no particular order. But what's interesting about microbes is that they will actually focus on one carbon source before moving on to another. So for example, you can kind of see in these growth curves, if we're growing on glucose and xylose, for example, we have the growing on glucose stop and then the growing on xylose. And this makes sense if you remember back to like the lack operon and some of the molecular circuitry that needs to happen, because it's not always on. Instead, in order to be able to grow on xylose, for example, we need to turn on new genes, and that can take some time. So this is just something to understand about microbial ecology, is that they will consume different carbon sources in a sequential pattern, and that can result in separate growth phases, and that is based on the underlying molecular circuitry and the need to turn on machinery to degrade a new compound. And then, at a high level, in the human gut microbiome, we know that diet can change how our gut microbes are metabolizing things. This is another study by my advisor, and he had, in this study, volunteers ate either a plant-based diet, meaning they had no meat, no milk, no eggs, or an animal-based diet, meaning they essentially only ate meat and cheese for a week. And so in this study, they were able to see that the gut microbes produced different chemicals based on the diet. So if they were on the plant-based diet, their microbes produced more acetate and butyrate. But people on the animal-based diet produced more isovalerate and isobutyrate. And these are called branched-chain amino acids, and we know that they are associated with protein intake. At the RNA level, so the transcripts is another good way so we can look, directly at what products are being produced at some point, but it's also useful in many cases to look at what transcripts, what RNA there are. And so here, you don't need to understand what these specific molecules are, but just you can see that like certain types of transcripts were seen at higher abundance in the animal diet. So what we eat affects our microbiomes in terms of not only which microbes are there, but also what they are doing. The other thing I want to talk a little bit about kind of how long-term, how metabolism can change. So we know that microbes can evolve over time based on their environment, their DNA can change. But these changes, one thing that's exciting and also potentially a little bit scary about microbes is that they can change really rapidly in ways that humans, for example, can't. And so there's this concept of horizontal gene transfer. First of all, what do I mean by horizontal? So vertical gene transfer is essentially, a mother cell to a daughter cell. It's transmitting the DNA vertically based on the lineage, the parental lineage. Horizontal gene transfer is where microbes that aren't related to each other can transmit DNA. And that can be done through a few different ways. The first here is transduction. This is carried out by a virus, such as a bacteriophage. So a bacteriophage is just a type of virus that infects bacteria. And so the phage can infect a cell. And we talked about how viruses sit their DNA into a cell, and then they kind of need to reassemble into new particles to then go infect new cells. During that reassembly, the bacteriophage can take up some of the host DNA, and then when it infects the next cell, it can transmit it. A great example of this is with cholera. So, in cholera, there's a toxin called the cholera toxin that is responsible for a lot of the disease symptoms. And that toxin is actually like not originally from cholera itself, but it's from phage. So a phage had that toxin. And so only cholera bacteria that were infected with that phage are actually going to cause this really bad disease because they have this toxin. Another example is conjugation. So yesterday we talked a little bit about plasmids. They're these like smaller circular pieces of DNA. And one of the benefits of having them is that they can be very mobile. And so in conjugation, two bacteria can literally link up and transmit DNA like that. And so that's one way plasmids can be transferred. Another final way is through transformation. And then this is basically where bacteria just pick up random DNA that's floating in their environment. Usually that's going to come from dead bacteria lying around. This is very useful in the lab because we can make bacteria competent there's a few different ways. One is by like heating them, another is by shocking them electrically. So it kind of makes them open up and take up foreign DNA. And so then we can put plasmids into bacteria. For example, GFP, green fluorescent protein. If we want to make bacteria glow green, we can do that. So just basically, important to understand that this is one way that the bacterial genetics can change. And, it might be useful to have certain plasmids if you are, in a certain environment, but other times, bacteria will just spit them out. They're very transient. They can be gained and lost depending on what you need. And then, I talked about this can be scary. One of the reasons is because antibiotic-resistant genes are often carried on a plasmid or other mobile element, and they can be shared between bacteria in these ways. So antibiotic resistance can be spread throughout a population. The very last thing I want to talk about today is that microbial metabolism can affect host metabolism. So again, in the gut microbiome, what our microbes are doing affects our health. This is a 2018 paper that I really liked from the scientist Elaine Chao out in California. And some of you may have heard of the ketogenic diet, which is essentially a diet that is, you eat a ton of fat and basically no carbohydrates. And where that came from, some people are using it for weight loss, but where it came from originally was in treating patients with seizures. And so we knew that something about this diet made people have fewer seizures. And this paper showed how the particular diet changed the gut microbiome, including their activity of producing certain compounds. And that changed which compounds were in the large intestine, changed which compounds were present in the blood, and then in the brain, and then how that led to changes in seizure susceptibility. So, you don't need to know what all of these chemicals are, but just what's important to understand is that in humans or other animals, what compounds our microbes are producing is going to affect what the rest of our body is doing as well. So diet modulates microbial metabolism, can modulate human host metabolism. And then in my own work, I just want to briefly share one of the things I'm working on is a study seeing whether increased fiber intake can affect human cognition. So the study looks like this. It's a 3-week study. And during week 2, we have participants eat these custom snack bars twice a day for 5 days. And those snack bars either have 9 grams a day of inulin, which is this type of dietary fiber I talked about, or maltodextrin, which is, the human body can break it down. It's basically short chain starch. So that's essentially a placebo that we expect to see no effect with that. And so then week 1 and week 3 are just kind of before and after, establishing like what the baseline state of the microbiome looks like for each person, and then seeing if it changes. And what we're looking at here is whether eating a lot of fiber, basically, over a week can affect someone's cognitive ability. These are screenshots from a cognitive test that we give to participants. So for example, in this one, they have to just click on squares as fast as possible, so staging reaction time. And similar kind of models in some ways after this study, we're trying to look at metabolites in the stool, in the saliva, in the blood, to try to understand if we do see changes, what changes in host metabolism are leading to these super high level things like cognition. I don't have the results yet from that. Maybe in a year or so, but that is a study I'm excited about. That is everything I have for today. We're right at about 50 minutes. Anyone have any questions for me about anything we talked about or anything at all microbiology related?
Hello, Mr. Jeff.
Yep.
Okay. You talked about microbial metabolism affecting host metabolism.
Yep.
I wanted to ask, the metabolism of the gut microbes affect other parts of the body? Like if we have the gut microbes and the skin microbes, does their metabolism affect other parts? Like can the gut metabolism affect the metabolism happening in the skin of other parts of the body?
Yes. So what I talked about was the connection between the gut and the brain. That's called the gut-brain axis. We are increasingly seeing that gut microbes really do affect all parts of the body. So you'll see papers talking about the gut-muscle axis, the gut-bone axis. So yeah, through various mechanisms that kind of remain to be understood. This is where you, like future microbiologists, can work on some of these things. We don't really understand how it all is connected, but yes, it does totally seem like there is some effect on like pretty much every part of the body.
Thanks, Mr. Jeff.
Any other questions?
Yes, Mr. Jeff, you said alcohols are microbial waste products. Can you please throw more light on that? Thank you.
Sorry, what is your question about ethanol?
You said alcohol are microbial waste products.
Yes.
Can you please explain further?
So I guess when I say it's a waste product, what I mean is that it doesn't really do anything for the bacteria. It's just kind of, so basically like they have molecules like sugar and they just want to break the bonds. And by breaking the bonds, you sometimes are left with these smaller molecules. Ethanol is one of them. It's not useful for the bacteria. In fact, if anything, it can be toxic, because if you have too much ethanol, the bacteria or yeast, whatever, will die. So at a certain there's a certain tolerance, and that's why, for example, like we can make beer or wine that has a relatively low percent alcohol, but you couldn't have microbes make something like vodka or hand sanitizer just directly on their own, because they're going to die in the process. You need to distill it, which is basically get rid of some of the water and just kind of capture the alcohol that's there. Another, what shows that this is like a toxic molecule to microbes is the fact that hand sanitizer is typically made with alcohol. Usually 70% seems to be a pretty optimal concentration where it kills microbes. Does that answer your question at all?
Yes, thank you very much.
Great. Anybody else?
Mr. Jeff. Mr. Jeff, can you hear me?
Yes.
Okay. I was talking about, you talked about bacteriophages attacking bacteria, the phages, viruses that attack bacteria. Now, when they do attack bacteria, do they cause any harm to our skin, to us, or anything like that? Or just specifically attacks the bacteria?
Yeah, that's a great question. Mainly, and it's funny to me because, like, to me, bacteriophages look like kind of the scariest of all viruses, but we generally don't have to worry about them. That's a great question of whether they could, adversely harm our health by hurting our gut microbes. And I'd say I'm sure it's possible. I haven't heard anything about it. I think it's an area that's relatively understudied. Again, you could work on this. But one thing I thought was funny is, I don't know if there's a market for this where you are, but in the US, there's a lot of dietary supplements you can buy in stores that are kind of unregulated. And so one of my lab mates, Zach, he found like a supplement, like a pill bottle that's just like phages. Like you can eat, and it's supposed to like get rid of some of your bad bacteria, but it's like totally unregulated. We have no idea if that's going to kill the good bacteria. And so we've thought about testing it in the lab before, but we haven't done it yet. So yeah, I'm sure there can be phages adversely affect your gut health, but I think there needs to be more studies on it probably.
Thank you, Mr. Jeff.
Yep.
Yeah. Good evening, sir.
Yep. Hi.
Good evening, sir. Yeah, my question is, what are endosymbiotic theory used for? Why are they used for? What's their function?
Like, why is it useful?
Yeah, about endosymbiotic theories.
I'm sorry, I'm having some problems with audio. Could you repeat your question?
I mean, what are endosymbiotic theory used for? Theory. What is the implication? Like their uses, the applications.
That's a good question. Unfortunately, I don't know that I have a solid answer for you. I think it's, my understanding of it has been more from just the understanding of the origins of multicellular life. I mean, I guess that's one of the big implications is that, it perhaps allowed us to transition from single-celled organisms into multicellular organisms, because we had within our cells these other smaller cells that could do some of the hard work of energy generating for us, leaving the bigger cells to focus on other processes. I think maybe one other place where it's important to understand about the mitochondria are there are certain diseases in humans, inherited diseases of the mitochondria. So you inherit your mitochondria from your mother always, not your father. And so understand, because, there's an egg, the egg has DNA and a mitochondria, and then the sperm just have DNA, they don't have mitochondria. That's why you only inherit them from your mother. So understanding a little bit about how mitochondria work, perhaps using bacteria as model systems, might help us better understand some of those inherited diseases of the mitochondria.
Okay, thanks, sir.
Yep, no problem.
I have a question.
Yep, go for it. Sorry, could you repeat that? I didn't hear the most recent question. Maybe if you want, you could type it in the chat. Okay. Well, if there's no other questions, then thank you all for your attention over the past few days. I will share the recording of this this past hour, and I'll share all my slides and everything. And if you ever have any other microbiology-related questions, feel free to reach out to me by email or Twitter. I'll provide that information as well. Yeah, thank you all.
Thanks, Mr. Jeff.
All right.
Thank you, sir.
Bye. Stay safe, everyone.